So girls, while studying the non-competitive type of inhibition, we come to know there is a allosteric modulation also. That means there is a change in rate of reaction due to binding at the active site. That means there can be two types of inhibitors. When it increase the rate of reaction like this, they are called as positive modulators and that is called as activation. Here we can see that enzyme inhibitor, what it does, it has the same allosteric site. That means this is the enzyme, this is the substrate active site, but enzyme inhibitor, it has a different binding site other than the substrate. Like substrate will come here and bind it, but no. Enzyme inhibitor, what it will do, it will bind at the different active site like this, and this is an enzyme inhibitor, this is the active site, and there is a change in shape of the enzyme. So when there will be change in shape, then substrate cannot bind here, right? So there will be total conformation changes and there will be no product formation, because it decreases the affinity of the enzyme for the substrate. Like this. This is called as allosteric inhibition, where there is decrease in the rate of reaction due to change in the shape of the active site. Clear? Example is, we have glucose 6 phosphate. It inhibits the hexokinase enzyme. What it does, it binds at a active site different from the substrate. Inhibitor has this shape and this is the binding site of the substrate, but enzyme binds here and it forms this complex, so that substrate cannot bind here and there is no product formation. Okay? Then second is when enzyme acts as an activator or there is increase in the rate of reaction. We have the example of fructose 1,6-diphosphate. Here when it binds here, when it binds, it is called as activator. This is the active site of the enzyme, this is the enzyme, and this is the activator. We can call it as activator or positive modulator. Positive by positive, because it will increase the rate of reaction. You can see here it is binding here and this is the active site, here the substrate is binding, this is the inhibitor, but still they are forming the product. That means it is a positive modulator. There is no change in the shape of the active binding site of the enzyme. But in negative modulation, there is change in shape of the binding site, so there is no product formation. Then we have another type of inhibition which is called as feedback inhibition. Now, before understanding the feedback inhibition, you should first understand how the synthesis of cholesterol takes place. Here the acetyl coenzyme A, it reacts with beta hydroxymethyl glutarate coenzyme A in the presence of this enzyme HMG-CoA, hydroxymethyl coenzyme A reductase, and then it forms a cholesterol. This is the synthesis process. But when there is excess cholesterol in the body, in our serum, lot of cholesterol accumulates, then what cholesterol does is, it inhibits this enzyme so that there cannot be further synthesis of cholesterol. Like when we have to regulate certain biochemical reactions where excess cholesterol will be harmful for us, it will cause problems for us. So in that way, cholesterol will inhibit this enzyme so that excess cholesterol will not be synthesized and only normal values will be synthesized. So after restoring the normal level, the cholesterol blocks this enzyme reductase, stopping the synthesis of cholesterol. This is called as feedback inhibition. That means the product itself inhibits the enzyme and stops the synthesis. So thank you so much.